So today I'm going to talk about DNA transcription, translation, and mutation. So starting off with nucleic acids, there's your DNA and your RNA. So DNA stands for deoxyribonucleic acids, and RNA stands for ribonucleic acid. So they both consist of three main parts, which are five carbon sugars, phosphates, and nitrogen bases. So DNA has four nitrogen bases, which are adenine, thymine, cytosine, and guanine. So DNA is in a 3D alpha helix shape, but I have a 2D model here that represents DNA. So DNA is double-stranded, and it's got its leading strand and its lagging strand. So they both have a backbone, which contains sugars and phosphates. The leading strand contains a sugar and a phosphate in a group called a nucleotide in between each base. And it continues its way all the way down to the bottom of the strand. And it's the exact same thing with the lagging strand. However, because it's backwards and anti-parallel to the leading strand, it's phosphate to a sugar, and so on and so forth. So in terms of bonds, adenine binds with thymine and cytosine binds with guanine. So because cytosine and thymine are one-ringed bases, they're called pyrimidines, and adenine and guanine are two-ring bases called purines. So this is where transcription comes into play. So transcription is essentially when the alpha helix shape unfolds itself and creates another strand called the mRNA strand, which replicates the lagging strand of DNA exactly, except for the thymine is replaced with uracil instead. So the mRNA strand contains codons, which are arranged in groups of three. So what happens is this strand of mRNA ends up leaving the nucleus, and it makes its way over to a ribosome that's just floating around in the cytoplasm. So as it enters the ribosome, it finds its start codon. So let me show you. So there is one start codon and three stop codons. So once your mRNA reaches the ribosome and finds that start codon, it starts to, tRNA ends up making its way over to the group of three and ends up pairing its anticodon with a codon to create its first amino acid. So the start codon is AUG. So we've got this one representing, AUG. So essentially the tRNA, the transport RNA, comes in and drops off this first amino acid to start a chain. So it moves to the next group of three. So we've got GAU. So that's this one. And it attaches itself there. And then it does the next three. So we've got UUG. And then we've got the next, which is GCA. And then we've got UGG. And the last one is UAG. So UAG is like one of the stop codons. So basically once you reach one of the stop codons, the protein stops making and your amino acid is finished. So it ends up making a shape of some sorts, and it transports itself to the next step. Yeah. So the next thing I wanted to talk about was genetic mutations. So mutations are caused by a number of things. One of them is just a mistake in the creation of your DNA. And then other mutations are caused by things like chemicals, UV radiation, things like that. Anything can alter your DNA, really. So there's two types of mutations. There is point shift mutations and frame shift mutations. So I'll demonstrate how that works real quick. It's quite simple. So in a point shift mutation, it's really just the replacement of a base. So essentially, removing one base and replacing it with another. A lot of the times this does not affect it. It's really considered a silent mutation because it's still a mutation, but it's nothing that can cause an issue. Because sometimes when you change just one base, you're still like technically within that range of amino acid. So if it's the exact same, or sorry, I'll show you. So like, let's say, you know, you're within this range here, any of those, like, it's not going to make any difference. So a frame shift is when you actually insert a new base or completely remove one. So let's remove that. So this will mess with your genetic code. When this happens, that's when you get genetic issues, of course, such as, let's say, Down syndrome, you know, things like that. It's the insertion or deletion of an amino, or sorry, of a base. Yeah, so that's basically it. I'll just show you my diet. I have a bit of a mess here. But we have, this just represents which colors are which bases. This is a picture diagram of how the nucleotides work with the leading and the lagging strand, as well as the bonds between adenine, thymine, cytosine, and guanine. Then you've got talking about your mRNA, how uracil is replaces thymine, and that it's single-stranded with A, U, C, and G, and contains 3 codons, your types of mutations, and then this is the amino acids and their codons. So you can see all that there. Yeah, that's basically the difference in mutations and how transcription and translation works in the simplest form. So yeah.